Well, hello, everyone, and welcome to Dr. Alain Rook. Thank you for joining us today, Dr. Rook, and for our fireside chat. It's really lovely to have you with us.
Thank you very much. It's a pleasure to be speaking with you, Susan.
And just so for those folks who may not know of you, you've been with the University of Pennsylvania as the director of the Cutaneous Lymphoma Service for quite a while, and been in the field of cutaneous lymphoma, both clinically and doing research for many, many years. And it's really wonderful to have you be here with us so we can introduce you to folks that may not have known you personally or have seen any of the presentations, many that you've done for us. But I'm sure many people recognize your name on a lot of the research papers and things like that. So let's get started. I'm really curious from your perspective. You've been in the field for a long time, but cutaneous lymphoma is not top of most people's areas of interest. I'm really curious how you got into the field of cutaneous lymphoma, and what attracted you to join a rare disease that pretty much not many people have heard of.
Well, it was rather a slow migration into the field. I was actually a very late comer to the field of dermatology in general. I was nearly 40 when I started in dermatology. I've trained previously in internal medicine and as a nephrologist, focusing on certain complications of renal transplantation, particularly the viral. And I spent many years at the National Institutes of Health studying viruses that occur quite frequently following kidney transplantation, namely cytomegalovirus, which is one of the more common infections and one of the more problematic infections to occur following renal transplantation. And at that time, when I started at the NIH, those were the early days of the AIDS, we should call it the AIDS pandemic, because it was a pandemic in those days. And I began to study viral issues in relationship to AIDS. And I studied AIDS and cytomegalovirus for nearly 7 years while I was at the NIH, and became very interested in the skin as a window to systemic disease, and decided to pursue my 4th training program in dermatology. And because of my background, I was appointed director of the Photopheresis program in 1987 at the University of Pennsylvania, which had developed at that time one of the first 3 active photopheresis programs in the United States. And it became busy very rapidly. Our clinics became busy very rapidly when it was learned that we had this program available for patients with leukemic forms of cutaneous T-cell lymphoma. And it wasn't long before we began using photopheresis for other clinical indications, for graft versus host disease and cardiac transplant rejection. And one thing led to another.
Wow. And here you are. And we're so grateful that you actually wiggled your way into the cutaneous lymphoma world, maybe from an odd way to get here. But, wow. So photopheresis, I didn't realize it was used back in 87. And, a little bit of my history, being diagnosed in 1991. And we're both here in Philadelphia. It wasn't widely used, and there probably weren't a lot of centers then, as you said, back in that time frame. there weren't a lot of options. There weren't a lot of specialists in cutaneous lymphoma. There weren't a lot of treatments. What was it? What was the landscape like for patients that were diagnosed in the late 80s or early 90s?
The therapeutic landscape was far from what it is today, in the sense that there were many fewer therapeutic options back in the 1980s. In fact, photopheresis and the very first photopheresis device that we used beginning in the mid-80s was quite an effective device for leukemic patients with cutaneous T-cell lymphoma. Yet there were few other options that we regarded as effective immunotherapies in those days. And quite frankly, when I started in the field, I was very pessimistic about what the outcomes would bring for most of our patients. And very early on, we decided, and I decided, coming from the background of an immunotherapeutic approach for immunosuppressed patients with AIDS, we started our approach to immunotherapy of both early stage and advanced stage cutaneous T-cell lymphoma right at the inception of my beginning in the field. And so we began to develop an enormous experience using different interferons to treat patients with cutaneous T-cell lymphoma. And in fact, we started combination therapies very early on in the mid-1980s, combining interferons with photopheresis, and then adding on top of that various retinoid compounds, such as isotretinoin, originally known as Accutane, etretinate, which unfortunately is no longer available for treatment of cutaneous T-cell lymphoma or any other indication. And we noticed enormous success using this approach. And we began to push very, very hard, both from the development of clinical protocols and from a laboratory perspective, where we decided we were going to focus on not only what is the overall picture of immune dysregulation in patients with cutaneous T-cell lymphoma, and how could we alter their various levels of immunosuppression by applying various approaches related to immunotherapy. And it was a very successful way to go. And numerous new approaches evolved from those early days of using interferons with retinoids, using things such as toll receptor agonists in clinical trials, some of which are well known to patients, including one that is FDA approved for other indications, namely imiquimod, but there are more potent drugs in the same class that are in clinical trial. Mogamulizumab could be considered a form of immunotherapy because of the way it alters the abnormal T cells. And there are other such approaches in the field.
Oh, so, and what's fascinating to me as I'm listening to you share this history, it really wasn't that long ago, right? So, I mean, when you think about it in terms of available therapies and things, that was what, 30 years ago. And we still use that same philosophy and a lot of those same therapies in combination. And I think, when I talk to patients and they're trying to understand the treatment landscape, it really truly is an art. And you were one of the original artists, if you will, in looking at how do you manage this very complex disease and how it interacts and what combinations of things could be the most beneficial. So many patients today are still on that sort of that triple therapy with a retinoid plus the photopheresis and perhaps another therapy. So that's amazing. And
well, I was going to say, I don't consider myself to be so much of an original thinker, but there were two very important publications that appeared in the medical literature right around the time I was commencing my career in dermatology. And one was from the NIH, establishing the fact that very aggressive therapy with multi-drug chemotherapy fared no better than low-dose methotrexate, topical chemotherapy, and radiation therapy. And in fact, patients seem to have an even better outcome with the less aggressive approach, and leading to fewer infections. And infections are a problem for patients who are on very aggressive chemotherapeutic approaches. And then there was a subsequent manuscript published by Elise Olson from Duke in what I consider to be a seminal trial using interferon alpha to treat cutaneous T-cell lymphoma, using it in several different doses and outlining certain hallmarks of patients that were likely to respond. So as soon as we digested the paper from the NIH, and I believe it was Paul Bunn was an important author on that paper driving the research, we shifted our approach to think about, well, chemo doesn't do the job. We need to think about immunotherapy. But I would like to think that we were among the earliest thinking about immunotherapies long before things such as PD-1 hit the literature. And now we have drugs like Keytruda, which is pembrolizumab, and others for treating cancer. And in fact, we use it now for cutaneous T-cell lymphoma, where cells express PD-1, which is a molecule that slows down the immune response. So these observations have led to opening the field to so many new immunotherapeutic approaches for our patients. And now we're moving into the era of targeted therapy, where we know that the malignant T cells, while they don't necessarily have a unique molecule on the surface that has yet been identified, they do have high levels of certain molecules that can be expressed on normal T cells, but usually not at the levels at which they're expressed on the malignant T cells. And one example is the molecule CD30, which often is expressed in tumor stage cutaneous T-cell lymphoma. And there now is a wonderful agent to target CD30 positive cutaneous lymphomas, and patients who have been on it know the name well. It's referred to as brentuximab, which immediately targets CD30, and it has a toxin that is engulfed or is ingested by the abnormal T cells that kills them. It's a very effective compound. It does have some nuisance side effects of peripheral neuropathy. And now with mogamulizumab, it targets the molecule CCR4, which is very highly expressed on the malignant T cells in the blood. And mogamulizumab is a very, very effective agent for Sézary syndrome and other leukemic forms of cutaneous T-cell lymphoma. So we're now in the era of targeting things, and there are even newer approaches that are being developed at the present time.
Well, it's really exciting to see how where you started, and I think that's where your background in working at the NIH and AIDS and immunosuppression really has had a huge benefit and an impact in cutaneous lymphomas because of looking at it in in that way and looking at the T cells that are part of our immune system that have gone awry, that are causing the cutaneous lymphomas. So, wow. And again, it hasn't really been that long. So let me ask you a little bit about, you mentioned Dr. Olson, Elise Olson, who's also been a leader in the field. And, I'm guessing back when you began in cutaneous lymphomas, did you know other clinicians and researchers? How did you connect with Dr. Olson or some of the other folks and clinicians who were doing this kind of work, so you could share and because there were no clinical protocols really at the time, right? How do you treat patients?
By extensively perusing the medical literature. I did know Richard Edelson very well, and Dr. Edelson is considered one of the key inventors of the initial photopheresis device. And I'll stress that initial photopheresis device worked better. We're now using this third generation photopheresis device. And the third generation device is not a bad device. It is an effective device, but the responses, the clinical responses generated by the first generation of the photopheresis device were eye-opening. 25% of our patients with Sézary syndrome went into clinical remission with that treatment only, and some of them never relapsed. And others who had no experience with photopheresis couldn't believe it. They failed to believe it. In fact, at one scientific meeting where I was presenting our results, I was shouted down at the podium, and I was told that it was fraudulent data and that there was a 25% spontaneous remission rate among patients with Sézary syndrome. Well, patients with Sézary syndrome have about a 0% rate of spontaneous remission. So we were seeing something very important. But unfortunately, when that first generation went off patent, Johnson & Johnson developed a new generation device, and all of our patients started getting worse at that point. So it was somewhat distressing. So when the current generation came along, it was refreshing to see that once again, we had a device that was producing benefit for our patients, but certainly not to the extent of that first UVAR device. And so Dr. Edelson and I have discussed these issues over the years, and he's been a refreshing colleague who has an enormous understanding, along with Michael Girardi, who's in the same program, of how these things work mechanistically. And of course, we're always interested in mechanisms and how do we make these treatments better. And as soon as it became known through work of Madeleine Duvec and some other European investigators that photopheresis induced so-called regulatory T cells. And what those T cells are, they serve as natural brakes on the immune response so that the immune response doesn't go haywire and attack our own bodies. And in fact, in autoimmune disease, there often are malfunctioning regulatory T cells that don't keep the immune response under control. In the case of photopheresis, they generate regulatory T cells, and as soon as we learned that, then it became all the more important for us when we were treating cutaneous T-cell lymphoma to understand that we had to activate the immune response further so that they could actively kill their tumor cells. And that also inspired us to use immune stimulatory compounds. And so we moved from things like interferon alpha to interferon gamma, and we are the most frequent users in the world of interferon gamma in our program. And then we did clinical trials with a compound called interleukin-12, very effective for early stage cutaneous T-cell lymphoma. and when combined with radiation therapy, was very, very effective in inducing superb responses. And now there is a company that is making, I have no relationship with that company, but it's called Kodiak with a C, and they are doing a clinical trial using what's called exosomal interleukin 12. And I certainly wish them well, because there's no doubt about it, and I think we would all agree that having as many immune stimulatory therapies available for these patients as possible would be highly beneficial, because we know that many patients will become resistant to interferon and not respond to photopheresis. So we need other things to add so that their immune response can be maintained against their tumor.
And that's always been the challenge. I wrote that company name down because I need to reach out to them and learn more about what they're doing. As I've participated and listened in the scientific meetings as well, the challenge is the resistance, right? You might find an approach that works for a patient and it's working, and you're going along and you're like, great, this is great. And then all of a sudden it stops working. And, our toolbox, unlike other cancers that may have more of a black and white treatment pathway, ours gets complicated, and we need more and more agents, like you said, to be able to almost mix and match. And when patients become resistant to a specific treatment regimen, there's something else that you can, put into the mix. But it sounds like, go ahead.
The issue you're touching upon is an extremely important issue, and is the basis for us using what we refer to as multi-modality approaches for advanced cutaneous T-cell lymphoma. And the basis is, we know from all cancers, and not the least of which is observed with cutaneous T-cell lymphoma, is that the cancer cells can outsmart one therapy and a second therapy by developing resistance. And we know in cutaneous T-cell lymphoma right from the start that there can be clonal variants. And we have seen it, and we've reported on it, and we've seen a patient, for instance, go into clinical remission on interferon, bexarotene, and photopheresis, and 6 months later relapse, and we could show that it's a new clone that no longer responds to the therapy. And we've published that in the Blood Journal back around 2010. And so we know this occurs, and that serves as the basis for a multimodality approach in an effort to try to prevent the cells from outsmarting our therapies. So particularly for advanced disease, we think about, if we're going to go straight to something that's not targeted therapy, such as interferon and bexarotene, I don't like to, for advanced disease in particular, to use one agent alone, because we've seen time and time again that the clone will outsmart the treatment. And we recently heard, for instance, Michael Cutaneous from Stanford present at the Society for Investigative Dermatology his experience with JAK inhibitors. And, for autoimmune disease, JAK inhibitors are becoming the rage. And what they do is they prevent T cells from carrying signals from the receptor of the cell into the nucleus to generate the end product of the cell. And JAK inhibitors at initial going looked very promising for cutaneous T-cell lymphomas. But he showed some excellent examples during his talk of how the clones develop mutations in their JAK proteins, which are JAK is short for Janus kinase, he showed very clearly that the Janus kinases that had initially responded to the JAK inhibitor underwent mutations and no longer responded. So these cells are tricky cells, and the same observation, for instance, was made with melanoma. Certain anti-melanoma drugs were initially found to be very effective, and the patients had dramatic responses, and then they would relapse very quickly, and they would find that the drug no longer worked. But now, when they use a two-drug regimen to help the malignant cells bypass the drug, avoid bypassing the first drug, the combination becomes much more effective. So we need to be thinking about how we do that constantly for cutaneous T-cell lymphoma.
Right. So we have a really smart disease is basically the underlying premise. I think trying to explain that to a patient who is struggling, right? Why is this happening? Why is this not working is really hard, and why we continue to need new therapies. Well, let me ask you, you've alluded to a lot of exciting things on the horizon and some new approaches. What do you see happening in the world of research? I think even in my limited time in my role and being exposed to the researchers in the field who are doing great work, there seems to be more people, at the most recent World Congress back in 2020, right before we all got grounded, I think there were 400 researchers, clinicians from around the world coming to share data and information, which to me is so fantastic, right? That there's this global connection amongst researchers and clinicians sharing data because the disease is so challenging. And learning new things and understanding how the disease works, and then putting the best minds in the field together, right? To try to figure out what should come next. What's your feeling on where we are today?
Well, we've made enormous strides since the mid-1980s. But we're still at a very primitive stage. That's my conclusion. But there are many new things being developed. And you may know, and many of my patients know, that I've been working on a group of compounds called toll receptor agonists since about 2005. And we have published multiple studies over the years, both laboratory studies and clinical trials that support the notion that this group of compounds can be game changers for the treatment not only of early stage cutaneous T-cell lymphoma, but possibly for advanced stage cutaneous T-cell lymphoma, particularly if they're used in combination therapy, as I've just stressed. And what toll receptor agonists are, and patients know very well about one compound called imiquimod, which at times is not very effective because its bioavailability, or its ability to cross the epidermis into the skin so that it activates immune cells in the skin, is really quite low. So it has to be used frequently and for long periods to be able to generate a local immune response. But some patients are quite sensitive to it, and it produces some really significant disease-modifying effects in some patients. And I was seeing a physician from Israel yesterday who used imiquimod. Most of my patients use one or two of these little quarter-gram packages on a single spot or two spots. Well, he had quite a few spots, and he started using 10 to 12 packages a day all over his skin surface. And he went to see his local expert in cutaneous T-cell lymphoma in Israel. And I saw her at the World Congress meeting in Barcelona early last year. And she said, I'm very troubled. I think he has erythroderma and he's developing Sézary syndrome. Well, I contacted him and I said, why don't you stop it for about a month and let's see where things are going. And his skin was completely clear after that. So all the redness in his skin was his local skin immune response that was generated by imiquimod. And let me back up here. Toll receptor agonists are a group of compounds that bind to toll-like receptors on the surface of cells of our innate immune system. What is our innate immune system? We have been programmed, and right back to very primitive organisms, yeast have toll-like receptors to respond to environmental pathogens, to environmental danger signals, so that they can actively defend themselves immunologically. So that when we encounter bacteria, whether it's on the skin or in the blood or elsewhere, in the abdomen, the first thing that gets triggered is these bacteria bind to cells of the innate immune response through their toll-like receptors, and they rapidly generate fever because our innate immune cells make things like interferon and interleukin-12 and tumor necrosis factor. And guess what? All those things are good for cutaneous T-cell lymphoma. We know that interferon works. We know that interleukin-12 works. And so we started a trial rubbing a powerful toll-like receptor on the skin. And lo and behold, it was a game changer for our patients. I am sorry to say, though, that a phase 2 sponsored trial by a pharmaceutical company had some issues where quite a few patients developed severe inflammation in their skin, which meant they were responding to it, and they allowed too many to drop out. And now the pharmaceutical company has decided that they're not going to move into an advanced phase trial using the drug resiquimod. And that news was devastating not only to me, but I've actually had some patients start to cry who were in my phase 1 study who've had their lesions come back now 6 years later, and they were hoping it would be in the marketplace so they could restart it. I've had several patients who, just by putting it on 3 or 4 skin lesions, and despite extensive involvement in their skin, had their entire skin clear because it got well absorbed into the blood and activated circulating immune cells and triggered a generalized immune response against all of their lesions. So there are a host of other companies that are developing compounds like this, but it's going to be years before trials are ongoing for cutaneous T-cell lymphoma. I am working with a new biotech company that is developing both an injectable version and a cream version to be applied to the skin, but they're just at the early phase of development, and they're desperately in need of investors to be able to push their program forward. But I'm going to, work my heart out to help them get it to the marketplace, because these are very valuable treatments, not only for cutaneous T-cell lymphoma, but for just about every other kind of skin cancer.
Oh, devastating news on that. So I've been following that trial too, and was hoping that we would be able to move forward. And that's the challenge. Again, sadly, when you're talking rare disease and the cost of doing trials and so forth, I think one of the missions that I see for us as the patient organization is to educate patients, to let folks know about these trials, because we need to enroll people appropriately, of course, in the trial so that the data can be gathered, and assuming that you see the ultimate responses that you expect, that we can continue to move forward and bring these really impactful new therapies to the market. And then we have a whole nother issue of, once they're available, to actually be able to have people prescribe them in different countries, but that's a whole nother topic. And another reason why we're hosting this first international patient conference, because we're attempting to bring together the patient community around the world, let people know what's happening, and then also gather the feedback from the patients so that we can do our part in making sure that we can move things forward. Because, you know, and at the end of the day, it's all of us who are living with this disease that need new therapies and new treatments.
Another issue, the patient should not give up hope. There are many, many things in clinical development, and there are clinical trials going on here, and Dr. Ellen Kim is carrying the ball in performing multiple clinical trials with promising new agents. But one of the issues that has stymied ongoing studies has been that big pharmaceutical companies, although they have often given lip service to developing drugs for rare conditions, and cutaneous T-cell lymphoma is considered in the US an orphan disease because there are less than 200,000 cases a year. And in fact, there are probably at most only 50,000 cases total in the United States, with maybe 4,000 new cases a year. Big Pharma doesn't really want to invest in these conditions. And we've had several promising clinical trials sponsored by biotech companies. Having the biotech companies purchased by Big Pharma so that they can use these drugs for their own purpose for more common disorders. And then they've stopped the trials for cutaneous T-cell lymphoma. It's been very, very frustrating. That happened with recombinant interleukin-12. That happened with a compound called CPG, which is also a toll receptor agonist to activate innate immune cells. The company making those was purchased by a big pharma, and then they stopped our study, even though it was showing enormous promise for advanced disease.
Yeah, it's a challenge. I will say, the more I learn about kind of what happens in the background, you know, as a patient, I was never privy. I wasn't in the pharmaceutical industry or in the research world, so I had no idea. And as I've become more engaged and involved in learning about these things, I become more passionate, shall we say, about really bringing that patient perspective to the table and why this is so important, and sharing the reasons. And the other piece of that is we also have to collect good data, because regulators and other folks rely on data and not just anecdotal patient stories, which are important, but we need the data to back them up, and what's the real impact on someone's life. And when you put thousands of people, even though 50,000 might not sound like a lot in the grand scheme of the disease, but that's 50,000 individuals whose lives are impacted by
Absolutely.
living with this disease, right? So
don't want to diminish that for 1 second. You know, for 35 years, I have seen a lot of suffering, and we don't want to diminish that. And I have treated other rare conditions as well that are life-threatening, that involve the skin. And these issues need to be clearly addressed.
Yeah. Yeah. It's, and as I just need more people, but that's a whole nother topic of conversation and not for today.
Well, we've
Let
got
me,
them here. We have them here at University of Pennsylvania.
yes.
I started here 35 years ago, and we had a paucity of faculty working in this field. It was myself and Dr. Stuart Lesson, who is no longer practicing, who left Penn, and that left me as the sole physician running our cutaneous lymphoma program, which was growing rapidly. But I'm very pleased to say we have had this steady flow of young faculty members joining our program, and we now have 40 clinics a month for cutaneous lymphomas in our department, staffed by 5 full-time faculty, all of whom have specialized in this area. And so it's really refreshing to see what's coming up behind me now that I'm becoming an old fart.
Well, I will say, you know, and again, I'm very privileged to live in Philadelphia and be close to Penn, and it has been amazing. I think I really have to thank you for your dedication to mentoring and bringing in the next wave of clinicians. You know, we've learned recently of, let's see, I want to say at least 2, maybe 3 physicians who are in clinical practice for cutaneous lymphoma have decided to leave clinical practice and go into industry or do some other things. And, you know, we don't have people in even at every major cancer center around the country in the US. And then when you get out of the US, it's a whole different and more challenging issue, access to people who really know this disease and understand it and know how to treat it, and how to work with the therapies that are available, and to know the difference between.
I can tell you that experience really counts for something.
Yeah.
You know, I'm not a youngster, and I'm still learning new things about finally altering treatments, tweaking this, tweaking that for some very difficult patients. Even in my old age, I continue to make new observations in regard to tweaking things. And as I move to the next stage of my career, and I'm becoming less active in clinical medicine, I almost don't want to give up my difficult patients, because I'm worried all these little tweaks that I've learned in more than 3 decades of dealing with this very challenging condition those issues may not be met, but I have enormous confidence in my own colleagues here.
Yeah. Yeah, we've been so privileged to tap into many of them. Dr. Ellen Kim was our first chair of our medical advisory council as we revamped it a couple of years ago, and we're so grateful for her knowledge and expertise in getting that component for us off the ground, and all the other folks that are kind of now falling in line behind her. It's hopeful. I mean, from a patient perspective, you know, I was originally seen by Dr. Eric Vonderheide, and who was one of the other, I would say, almost early researchers in the field. And, you know, but he retired eventually after being my physician for 15 years. And then I ended up with Dr. Stuart Lesson, who then also retired and left clinical practice. So it's like, wait, guys, I still have the disease. Come on. But but I'm lucky. You know, I live in an area where we have a depth of knowledge. And if my disease should progress or get strange on me, I know I have somewhere to go. and people that know what they're doing. But not everybody has that option. And I think being able to hear your experience helps us to educate the patients as well, to know what kinds of questions to ask and how do you work with your physician, because this is like a long-term relationship. So I don't know if you have any thoughts on having been in a clinical practice, and obviously many of your patients have been with you for a long time, right? For other people, how they might approach working with their clinicians as they go through their process, and how do you have that kind of dialogue that is beneficial? Any thoughts on that?
That's the most difficult question you've asked me this afternoon.
Sorry about that.
I think that's a highly personalized issue. I can only say that I try to be as truthful as I can with my patients based upon my personal experience. And I will mention that a few times earlier in my career, I told patients that I thought they were going to be fine. And then sometime later, they were not fine. And of course, family members were really quite upset with me. And that too was a learning experience. And for example, I had a patient come to my clinic who was a physician who had a few plaques on his skin, not patches, but plaques. And he had just come off of type of chemotherapy. And I was too young to appreciate, first of all, that if you have plaques, your prognosis is not as good as those who have patches, and that those types of patients need to be followed very, very closely. And if there is worsening at all, for which they have a significant potential, they then need to be treated aggressively. And, you know, when I have a patient who has many plaques, I will often start them on combination therapy. So I learned the hard way. Secondly, if I have patients who aren't doing so well, I always encourage them to get a second opinion or even a third opinion, because an expression I often hear is, if you have 10 rabbis in a room, you're going to get 20 opinions. And so I encourage my patients to get other opinions. And often it's very worthwhile for them and for me to learn something new. And I like to learn something new. Fortunately, as I said, we have 5 full-time faculty in our cutaneous lymphoma clinic. And we also have an absolutely outstanding T-cell lymphoma oncologist here at Penn, who really has an encyclopedic knowledge regarding treatment of cutaneous T-cell lymphoma. So we get a lot of advice from him, and I often, early on, if patients have telltale signs that they're at risk of progression, I often send them immediately to the oncologist. And we're also fortunate that we have an absolutely outstanding group of skin radiation therapists who can do total skin electron beam for patients with advanced forms of cutaneous T-cell lymphoma. And in fact, you probably heard me present at the Barcelona meeting the effectiveness of low dose, that means 12 low dose treatments of total skin electron beam combined with interferon and photopheresis and bexarotene for Sézary syndrome. And we put a very high frequency of our patients into complete remission using that approach. And often the remission is sustained for years. And so it's good to know that we have those colleagues who can teach us something about how to do that therapy. Unfortunately, some
Go ahead.
colleagues at other centers really feel compressed for time, and they may not spend as much time talking to their patients and answering questions. It's always been a goal of mine right from the start not to feel like I was pushing patients through a mill. And that's one reason why I didn't go into private practice, where I would have earned 2 to 3 times what I've earned as a university professor, because I like talking to my patients and spending as much time as necessary so we're both satisfied that all questions have been answered. And so that's a very comforting feeling for all of us. And I think my colleagues here at Penn base their cutaneous lymphoma practice the same way. I know at some centers it doesn't work that way, and it can be very hard on the patients. So, you know, general dermatology gets 15 minutes a patient, and in some cases, if they're lucky, 7 minutes. For a new patient, I allow an hour for a cutaneous lymphoma patient. You won't find that at most places.
Yeah, I wish that were the standard instead of the oddball. But I think that's the other piece of, sadly, you know, patients becoming their own best advocates so they can leverage the time they do have with their clinicians. And I really appreciate you sharing about the second or third opinions. We get a lot of calls from folks, and they tend to sometimes be reluctant, you know, fearful that they may hurt the feelings of their physician, or that their physician will feel like they don't have the confidence if they go to get a second opinion. But, you know, I always encourage people to get a second opinion, which is the other reason why we do our best to keep the treatment centers who specialize in cutaneous lymphoma and the practitioners up to date on our website, so that people know who's closest to them and where they might be able to go to get that second opinion. And I also let folks know, you know, I always feel like I'm the fly on the wall at the scientific meetings, and it's so heartening to me to see the collaboration amongst the clinicians in this field globally. I work with other disease patient advocates with other kinds of diseases, and the stories that I hear in other disease states is totally different than the cutaneous lymphoma space. And I think the passion, the heart, the willingness to share, the willingness to mentor and teach a younger group of clinicians is so impactful. And from a patient perspective, I'm deeply grateful for that, because otherwise I think it would be more challenging, and it's already challenging just dealing with the disease.
So many wonderful physicians and scientists from all over the world, from every continent, who have an interest in cutaneous T-cell lymphoma, who want to collaborate. And I believe you're going to talk to 2 of the finest after you complete my discussion. Sean Whitaker from London and Dr. Ivan Litvinov from Montreal, who are 2 of the most outstanding scientists and physicians in the field, who are pushing the field forward. And they both have outstanding research programs and have accomplished quite a bit. And you can't find better people in the field. There are some outstanding therapeuticians that you will not be talking to as part of the patient meeting, but I'm sure during the meeting you will call out their names. People such as Yeon Kim, who, and Michael Codaduce from Stanford, and Michael Girardi from Yale, and Elise Olson we've mentioned. And there are so many others that I can't even begin to mention. So many from Europe who have accomplished so much, and from Japan and Australia. So the world is rich with investigators.
Yeah, and we'll see how I do, but I want to do fireside chats with a lot more people, because I think bringing your wisdom and the wisdom of the researchers and the clinicians and introducing you all to the broader patient population, I think really brings us all together, because we're all part of the same community, whether we're on the patient side or you're on the research side or you're on a clinical practice side. We are all part of the puzzle, and we can't move forward without the dedication of folks like you. So one last and final question: when you look into your crystal ball, what do you see, and what, if you could wave your magic wand, what might the future look like in cutaneous lymphoma for patients?
I think what's called precision therapy could overtake the field. And judging by what's going on at Penn, in research by Megan Lim, who is chief of hematopathology, and Kojo El Natoba Johnson, who is the chief precision officer at Penn, they are developing a major scientific program to address the changing landscape of mutations in advanced cutaneous T-cell lymphoma, so that early on they can identify cellular mutations for every specific patient, so that mutations can so-called be drugged with active drugs that are available immediately right from the start, so that these cells can be halted in their tracks. And they're also developing a platform of their own for early diagnosis for patients where the diagnosis could potentially be delayed. And as all of our patients know by experience, diagnosis is often delayed by many years because it's a rare condition, and the practitioners on the outside are not attuned to making the early diagnosis. So addressing both early diagnosis and druggable mutations for individual patients will be an unmet need going forward.
That's exciting, and sounds like I need to circle back to those two amazing people that we've had present and see where they are with their progress. So thank you so much, Dr. Rook, for joining us, and for taking the time to share your experience and your knowledge and your wisdom. And I know so many people will benefit just by listening and being aware of what's here. And we're all so deeply grateful that you've spent 35 years of your career in this crazy, bizarre disease of cutaneous lymphomas, and look forward to whatever you're up to next, and talking to you again soon, maybe at our next year's international conference. We'll see what happens between now and then, but thank you so much. It's been such a delight and an honor and a pleasure.
Thank you very much, Susan. The pleasure has been all mine, and going forward, I look forward to helping more patients with this condition. Thank you.